Of course, I get interrupted in my good morning at the moment that my phone bleeps. Yes, good morning. Here we are. This is Thursday, the final day of April, and today we're going to be doing a little bit more review. The review today is going to focus around cell organelles, and kind of like we did in the last review session, what I did was I kind of took bits and pieces from some of the other stuff and wedged them in there too. Now, on the checklist, a lot of people were having issues with what is the purpose of organelles? How do they aid a cell in its essential or necessary functions? Things like that. Today, we should be able to answer all of that by the end. So, any questions, please put them in the chat. I'll get to them at the end. And otherwise, why don't we press on? So, this was originally something that we went over very early, and hopefully from the learning objectives, it's relatively straightforward. Everything is about how things are going to interact or how they're going to provide a function. And cell theory is the basics. We're not going to worry too much about that. Prokaryotic and eukaryotic, though, you guys do need to know the difference from or of. Yeah. So remember, your prokaryotic cells are going to be basically limited to archaea and bacteria. Those are two of the three domains, which some people had issues. Remember, the domain division or identification of life has archaea for the really, really bizarre prokaryotes. Then it has our bacteria for the more recognizable prokaryotes. And then everything else falls under eukarya because everything else is a eukaryotic cell. So our modern form of classification is actually entirely dependent on what type of cell we're looking at. A prokaryotic cell or a eukaryotic cell. And the big thing to remember is that prokaryotic cells really don't have any organelles. They don't have a nucleus. They don't have most of the other things like a mitochondria that we would assume a living thing should have. They do have ribosomes, but they're very, very simple, very small cells. Because of that simplicity, they don't have the structures necessary for multicellular life. So while they can work together, and we saw that earlier this year talking about biofilms, talking about quorum sensing, the ability of bacteria to speak to and coordinate with each other, they can live with each other, but they can't form a multicellular organism that has specialized organs and specialized functions for those organs. Eukaryotic cells, on the other hand, we're far more familiar with because it's us. We've got all the bits and bobs in there. We have all the pieces, the different sacs that do the different things. So let's dive into then the eukaryotic cell and take a look at what the different organelles do. Now, organelle, the term came from the term organ. When we look at an organ in something like a human body, we're seeing a smaller part of it that has a specialized function. When we look inside a eukaryotic cell, we will find they have smaller parts with specialized function. So they took organ, they took cell, and they kind of slapped it together, organelle. Now, here are the most important two slides out of this entire thing. Why? Why do we have organelles? Why are they successful? Well, first of all, organelles exist to compartmentalize the cell. That's incredibly specific and incredibly important. If you remember going over photosynthesis and cellular respiration, everything is about that chemiosmotic gradient. We have to have those things separated so we can make that gradient so that it can diffuse, so that it can power the ATP synthase transmembrane proteins in order to generate the sugars in the case of photosynthesis, or the adenosine triphosphates in the case of cellular respiration. Compartmentalization is incredibly important. And even beyond that, we wouldn't want lysosomes floating around without the ability to keep their enzymes stored, contained, and safely away. So it's all about compartmentalizing an area so that different areas can do different things. and we can keep the dangerous stuff sealed up. The other major function that they have is to increase the surface area. Now, people asked about this on the checklist. Like, how does surface area affect living things and functions? Well, the key here, and we actually saw it at least at one point during the year, the key here is just to realize that a greater surface area means more chemical reactions. The more surface area you have, the easier it is for stuff to happen. Case in point, why do humans have chewing teeth? Why aren't we like a bird where we just pick up our food and just gulp it down whole? Well, it's because chewing something increases the surface area of that food. That then allows our enzymes, our stomach acid, our digestive juices to work on a greater area, increase the amount of chemical reactions, break it down faster, so that we don't need a 75-foot digestive system to get our nutrients. We can make do with a 30-foot digestive system just to make sure that it happens fast enough. And interestingly, even birds would not be able to survive on average by swallowing their food whole. They have something called a gullet, which they'll also eat pebbles, and then muscles will flex and it will push and squash those pebbles against each other and against whatever they just swallowed to mash it up and break it up a little bit. So even birds do kind of chew, just not in the way that we're familiar with it. So that's the key for organelles. Number one, compartmentalize to keep things separate. Number two, increase surface area for more chemical reactions. So let's start looking at our individual organelles. We'll start with the ribosomes. These are found in both prokaryotic and eukaryotic cells, and they are the only organelle that is found in both. Now, for some people, they really don't classify ribosomes as organelles. They really think of them more as structures. Because remember, a ribosome, we've now learned, is literally composed of just a piece of RNA and a couple proteins, and that's it. So it doesn't have its own membrane. It isn't compartmentalized. It isn't separated. It's just a little bit of a glob that does an unbelievably important thing. Hopefully, all of you remember that ribosomes are involved in the central tenet of molecular biology, where we have to take that DNA, turn it into the RNA, and then the ribosomes come in because we got to turn that RNA into a protein. So that is the key. Ribosomes are the site of protein synthesis. It is where we make our protein. They like to use the word polypeptide. We went over it not yesterday, the day before in review. Remember, that just is the fancy word for protein. Then we've got the endoplasmic reticulum, both forms, smooth and rough. The only difference is whether or not they have ribosomes on them. If they have ribosomes, we call them rough because they look bumpy. If they don't have ribosomes, we call it smooth because they don't look bumpy. Now, the rough ER is going to be a site for specific proteins, and what that means is you see how the ribosomes are kind of embedded into the membrane here? Because they're embedded into the membrane, we can control very particular proteins to get made here where they will then immediately be contained inside this membrane, inside this pocket, to once again compartmentalize and keep them away, keep them separate. It also provides a bit of transport just because you can push things along through there to get from one place to another. The smooth ER likes to make lipids. In particular, it makes membrane lipids. Membrane lipids that form not just the cell membrane around the entire cell, but also the membrane around just about every single other organelle. So, very, very important. Hopefully, you guys remember lipids are one of our four biological molecules. They are our fats and our oils, things like that. They have that amphipathic nature, which gives them their hydrophilic and hydrophobic sections, mostly because they're composed of this one glycerol that forms the backbone and then the fatty acid chains. And the glycerol is hydrophilic, loves it, it's polar. The fatty acid chains are hydrophobic, they're nonpolar, so the fatty acid chains want to move away from water, the glycerol wants to move towards water, and that allows us to create that very special plasma membrane or lipid membrane. They're linked together by these oxygen to carbon ester bonds. Polypeptide bonds were for proteins. Ester bonds are for our lipids. Then into the Golgi. Looks like the stack of pancakes. Now, this has a the name for these separate stacks are cisternae. The problem with that is that they're very, very similar to mitochondria and the Cristae that are inside of mitochondria. So watch out for those differences. I suggest you just look for an N. If it's got an N, it's the Golgi. If it doesn't have an N, then they're talking about the mitochondria. Now, their job is really just the package stuff. Things will come in on one side. You kind of see it with this little drop down below my pointer here at the bottom of the page, and it's going to move up. It's going to hit these membrane layers, and as it starts to go through layer after layer, each one will add something to it. Another layer. It acts like packaging and stamping, so that then this can get transported wherever it is that it needs to go. The Golgi has often been referred to as like the UPS center of a cell. Now, that coating is our lipid bilayer that you see here once again. The mighty mitochondria. Alright, this is our powerhouse of the cell. The one thing that everybody seems to remember from high school, despite the fact that arguably, while it's nice to know, it's probably one of the most unnecessary things for day-to-day life for anybody. Because understanding how this amazing organelle works is fabulous if you are like a cellular biochemist. It is much less useful to almost anybody else on the planet. Maybe a geneticist. So they have a double membrane. Why? Because we want to compartmentalize. We need to keep things separate so we can build up that concentration gradient to make things work. How is it important to its function? That compartmentalization and that chemiosmotic gradient. The inner membrane has all the folds. These are the Cristae, not to be confused once again with the cisternae of the Golgi. And we're not going to go over the entire process of cellular respiration because we already did. But keep in mind, it is super important for us to make ATP and to maintain the amount of energy that we require to stay alive. And of course, the famous saying. Lysosomes. Fun, simple, dangerous. Because they're literally just a membrane filled with a whole bunch of enzymes. And if those enzymes come into contact and spill out onto or around another organelle, they will destroy it. This is apoptosis. So, for instance, if a cell is infected with a virus and a natural killer cell comes up to the surface of it, detects that antigen on the outer coating of the body cell, it will then say, okay, lysosomes, open up. And the lysosomes in that cell will follow that programming, that message. They will open up, spilling their enzymes into the cytosol or the cytoplasm of the target cell, and they'll do what enzymes do. They'll kill things, rip them apart, break them down. You should remember enzymes. They're a very particular and very important kind of protein. Remember, you're going to have the active site. That's going to be the shape. Because it's a protein, you see all those R's? Those are the R groups that have interacted in order to form this very specific shape that will match a very specific substrate. Most enzymes, in order to function, will require some form of cofactor or coenzyme. It could be called either of them. You'll see that right here in the middle. And really, all we're saying is these are other things that are necessary to make the enzyme active. It would be very dangerous for you to have enzymes constantly cycling your body, constantly open to substrate level interactions. They might break down things that we don't want them to. So, being able to control them with a coenzyme or a cofactor that you only need to produce when you need it can be very, very useful to us. Think lacoperon. Now, for a lot of enzymes, they need bunches of stuff. They need basically all the things to come into contact before they're actually ready to become active and start breaking down the substrates. Remember, there are a couple of ways that we can interfere with this. We can do allosteric. Now, this can work either way. Maybe the allosteric activator comes in as a cofactor, as a coenzyme. It hits, causes a conformation change. Now the active site is ready and the two can bind, or maybe it does the opposite. We also talked about allosteric inhibition, where the inhibitor molecule binds allosterically. Remember, allosteric just means not the active site. And that changes the shape. In this case, it would change it from square to circle, and now the substrate can't bind. So that would stop the process. You can see it right here. A vacuole, once again, is just used to compartmentalize. We either want to bring in the food or we want to bring out the waste. Both of those will happen by using a vacuole. Vacuoles and lysosomes are incredibly similar. The only difference is in what they carry. Other than that, they are functionally identical at our level of science. Plants sometimes have a great big central vacuole because water is so important to them. The chloroplasts, we are not going to go over all of photosynthesis. We did that already, but chloroplasts, the specialized organelle that allows it, we talked about how you're going to have your little stacks of thylakoid here forming one grana inside and how that works. The absorption spectrum, we see green plants because they don't absorb green. They reflect green. They absorb orange and red. They absorb blues and purples, but not greens. And once again, remember, a major function of this is describing how the structure supports the specialized function. The mitochondria had the double membrane structure. Here we go again. Chloroplast, double membrane structure. So once again, the same reason, compartmentalize it. Our nucleus, the glorious leader, to steal from SciShow. This is the control center, holds all of our genetic information. And when we talked about cellular communication, this is where all those signals are eventually going to go. They've got to reach this point in order for anything else to happen. And we're done. Whew. So you guys should be able to make predictions about how interels would interact. Not too bad. You know what they do. You should be able to construct explanations based on your evidence and how they provide those essential functions. That's going to be either compartmentalization or surface area. Then you should be able to use models to look at basically the same thing. How can these specialized functions happen? Now, I also then included these that come from our biomolecules part, where you should be able to kind of describe how a biological polymer is made. Well, the only polymer we've covered so far is proteins. We did that last time. You should be able to explain how it determines the properties of a polymer. We did. Remember, how those R groups interact will determine the final shape and therefore the final function of that protein. And you should be able to use models based on it. Okay. It's a lot of stuff done very, very quickly, but hopefully it helps. Hopefully it will help prepare and give you that additional practice and repetition that you need. If you have any questions or anything else, please let me know. And otherwise, I will see everyone on Friday.